Welcome to AllAboutRx. I am so excited to have you here for today's explainer because we are tackling a topic that is absolutely foundational to clinical pharmacology, mastering membrane transport. Seriously, whether you're prepping for your big physiology exams or you're just trying to deeply understand how medications actually work in the body, it all starts right here at the cellular level. Okay, let's dive into this. Think about it for a second. How do vital molecules, nutrients, and life-saving drugs actually cross the cell's ultimate border? You see, the plasma membrane isn't just some passive wall. It is a highly secure border crossing that strictly dictates exactly what gets in and what gets out of our cells. Every single physiological process you can think of depends on this microscopic security system functioning perfectly. So here's our agenda for today. We're going to hit the basics, move on to unassisted transport, cover osmosis and tonicity, dive into assisted and active transport, and then wrap the whole shebang up with a quick recall summary. All right, Section 1, the basics, membrane fundamentals. The single most crucial property of the plasma membrane is that it's selectively permeable. The key takeaway here is that this is absolutely not an open door where just anything can wander in. It acts as a highly selective border, permitting certain substances to pass straight through while entirely excluding others. So how exactly does this selective permeability work physically? Well, check out the hydrophilic, or water-loving heads facing outward, and the hydrophobic, water-fearing tails facing inward. Because of this fatty lipid interior, lipid-soluble molecules can cross right through, completely unassisted. The exact same goes for very small particles, specifically those under 0.8 nanometers. Basically, if you meet those criteria, you get VIP access to cross the border for free. Now, when things do move across, they're governed by two main categories of forces. First, we've got passive forces. These move substances without a single drop of energy expenditure from the cell. We're talking things like diffusion and osmosis. Contrast that with active forces. These are the heavy lifters. They require the cell to actually expend its own energy, in the form of ATP, to force substances across. Keep these two distinct forces in mind as we explore the specific transport mechanisms. Moving right along to Section 2, unassisted transport, or passive movement. Here we're looking at simple diffusion in action. Simple diffusion is just the net movement of particles from an area of high concentration over to an area of low concentration. And the crazy thing, it happens purely due to random molecular motion. You don't need a motor, you literally just need a concentration gradient. And notice how this net diffusion continues until equilibrium is reached. Now, at equilibrium, that random movement is still happening, but the net diffusion equals zero. Now, Fick's law of diffusion dictates the actual math behind all of this movement. It's pretty intuitive once you look at it. If you increase the concentration gradient, the surface area of the membrane, or the membrane's permeability, you increase the rate of net diffusion. It goes faster. But on the flip side, if the molecular weight of the substance is heavier, or if the diffusion distance is increased, the rate drops. It slows down. So why does Fick's law actually matter to us? Well, let's anchor it to a real-world clinical application. Think about respiratory conditions. Pulmonary edema involves fluid buildup in the lungs, right? That directly increases the diffusion distance. Emphysema, on the other hand, destroys lung tissue, which decreases the surface area. According to Fick's law, both of these conditions will dangerously slow down the life-saving exchange of oxygen and carbon dioxide. It is simple diffusion gone wrong on a massive macroscopic scale. Section 3, Osmosis and Tonicity. Let's talk water balance. Osmosis is a very specific type of diffusion. It's defined as the net diffusion of water down its own concentration gradient across a selectively permeable membrane. Now, because water is polar, you might be wondering, how on earth does it get through that lipid bilayer so easily? Well, it crosses incredibly fast through specific, dedicated water channels called aquaporins. Guys, we are talking about 1 billion water molecules per second zipping through a single channel. It's wild. In this classic YouTube setup, take a look at side 2. Here we have non-penetrating solutes, meaning solutes that simply cannot cross the membrane. And because they can't cross, they effectively pull water toward them to try and equalize the concentration. So water flows into side 2, increasing its overall volume. This flow only stops when the opposing physical force, the hydrostatic pressure of that taller column of fluid, pushes back hard enough to perfectly equal the osmotic pressure. And that right there is dynamic equilibrium. Let's see how this actually plays out in the human body by looking at the fascinating effects different solutions have on red blood cells.
Tonicity is all about the effect a solution's non-penetrating solutes have on cell volume. Look at what happens to the red blood cells when they are dropped into these three very different environments.
Let's quickly summarize that visual data. In an isotonic solution, the solute concentration is equal to the cell, so there's no net water movement, and the cell stays a totally normal shape. But in a hypotonic solution, the solute concentration outside is lower, so water rushes into the cell, causing it to swell and potentially lyse or burst. No good. Conversely, in a hypertonic solution, the concentration outside is higher, so water is violently pulled out of the cell, causing it to shrink and shrivel, a process known as crenation.
And this leads us to a vital, absolutely non-negotiable clinical rule. IV fluids administered to patients must generally be isotonic, like 0.9% normal saline. If you give the wrong tonicity, you are going to cause widespread unwanted swelling or shrinking of your patient's red blood cells. It's a perfect example of connecting a microscopic physiological concept to everyday, real-world patient care.
All right, Section 4, Assisted and Active Transport. Let's look at the transport systems. For vital, larger molecules like glucose or amino acids, the cell actually provides VIP escorts. We call this carrier-mediated transport, and it's governed by 3 strict rules. First, specificity. Each carrier only transports a very specific substance. Second, saturation. There's a limited number of these carriers. And third, competition. Closely related molecules can actually fight for the exact same carrier spot. Honestly, you can think of these carrier proteins as highly specific, easily overwhelmed bouncers at an exclusive club.
Now here we see facilitated diffusion, which is a passive form of carrier-mediated transport. Notice that no ATP is required here whatsoever, because the substance is still moving downhill from high to low concentration. The carrier simply grabs the molecule, changes shape, and releases it on the other side. A major clinical link here is glucose transport via GLUT carriers. Insulin actually increases the number of these GLUT carriers in the membrane, which is exactly why a lack of insulin in diabetes impairs the cell's ability to take in glucose. Comparing facilitated diffusion to simple diffusion, look at the green line for carrier-mediated transport. Because of that rule of saturation we just talked about, the rate of transport eventually plateaus. This plateau is called the transport maximum, or TM. Once all the carrier proteins are fully occupied, the system is maxed out, and the rate just cannot go any faster, unlike simple diffusion, which just keeps climbing linearly.
Finally, we've got active transport. Contrast the passive carriers we just saw with this. Active transport moves substances against their concentration gradient, from low to high. It's literally like pushing a boulder uphill, and that requires energy. Primary active transport uses ATP directly. A classic example is the sodium-potassium pump. The cycle involves phosphorylation, where the carrier binds ATP's phosphate to change its affinity and force substances across the border. And just for context, there's also secondary active transport, which is powered indirectly by ion gradients, instead of burning ATP directly.
Okay, let's lock this all in with a rapid-fire review. Diffusion and osmosis, passive. Isotonic means no change. Hypotonic means swelling. Hypertonic means shrinking or crenation. Facilitated diffusion uses a carrier, but zero ATP. Active transport uses a carrier and always needs ATP to fight the gradient. And remember those 3 bouncer rules for carriers: specificity, saturation, and competition. Keep this quick recall checklist in mind to guarantee you are fully prepared to ace your physiology and pharmacology exams. I'm going to leave you with this provocative thought. How will mastering these microscopic transport mechanisms change the way you understand macroscopic clinical outcomes? The next time you see a patient with edema or you hang an IV bag, you aren't just seeing symptoms or fluid anymore. You are seeing Fick's law and tonicity at work. Thank you so much for joining me for this explainer. Stay curious, keep learning, and I'll see you next time.